Welcome to this webinar about biogenic nanoparticles and how to track them in biological samples. But before I start with the topic, I would just like to know you. I'm not sure whether you can see this. Okay, where we're doing our research and where we are living. So Oviedo is the capital of Asturias, which is in the northern part of Spain. And we have, well, a beautiful landscape. You can see here's the access to the Atlantic Ocean. We have beautiful beaches and also some cliffs. And what you cannot expect is that from 1 hour or half an hour from the beach, you will reach the mountains with an altitude up to 2,600 meters. So this area is completely different to the rest of Spain because it's very green. And this coast, for example, is called the Green Coast. And well, as I can just say, Asturias Paraíso Natural, I think this is completely true. And we have beautiful buildings. For example, here you can see a building from the 9th century, and here this is the patio of the University of Oviedo, which is more than 400 years old. And what is very, very important is food in this area, food and drinks. So we have excellent seafood, we have excellent meat, and the specific drink of this area is cider, which is poured in this way. Sports is also quite important, and since I've been in Asturias, this is almost 20 years, almost 20 years I'm celebrating now. Germany was not so successful anymore in sports, but Spain has powered a lot. And well, Oviedo is a very nice place because even on the street you can see some famous people. For example, here Santi Cazorla. If you're a football fan, you might know that he has been a national player for the Spanish team some years ago, and now he's still playing in Oviedo. Okay, let's come to the topic, which is principle about nanomaterials. And in order to define it, well, I found this old recommendation of the European Union, which is 15 years old, and it hasn't changed a lot. And basically it goes, it's very complicated to read and I won't do it, but it implies also the particle number concentration, and only particle concentrations that have at least one or more external dimension in the range of 1 and 100 nanometers are defined as nanomaterials in principle. However, I will keep it a little bit simple, and I will talk about nanoparticles just in the range, in the size range between 1 and 100 nanometers. So, sorry. Maybe you don't know, but we are surrounded by nanomaterials all the time because there's a huge number of manufactured nanomaterials that you can distinguish between in organic ones. The famous ones are the fullerenes or the carbon nanotubes, and the one that affects us a little bit more are related to inorganic materials. That can be metal oxides, that can be metals or quantum dots. And here are given some examples. And especially here, these metal oxides and the metals we are surrounded in our daily life every day. So somehow in this century, it is also called one part of the century is called nanotechnology century. The value expected, for example, for 26 is even higher, but in 25 was more than 105 billion US dollars. So you can imagine that there's a strong interest in many fields using these nanomaterials. And one of the biggest one is, for example, healthcare. So we are in touch with nanomaterials that are used for diagnosis or even for treatment. And just mention here some areas where nanomaterials are used in nanomedicine, nanoelectronics, of course, because of the different properties these nanomaterials have. We find a lot of food products in which we intentionally add some nanomaterials. Could be, for example, titanium dioxide to make some materials more white, can be silicon dioxide to improve the properties of the food products. But you find also In cosmetics, a lot of nanomaterials, for example, zinc oxide, titanium dioxide, or even elemental silver nanoparticles. So the question is, how do we synthesize these particles? And basically, we have two approaches. One is the bottom-up one, in which we have a certain precursor, and under specific conditions, we first produce some clusters which contain a few atoms of the specific element, and then under specific conditions, you form these nanostructures. The other way is, of course, coming from the other side. It's called the top-down approach, in which we come from powder or from bulk material, and by physical methods, we lower the size of the material, and then coming up to the sought nanostructures. Here it's a little bit summarized. We have possibilities to synthesize these nanoparticles by chemical reduction, co-precipitation, sol-gel methods. But if we have a look to the top-down approaches, we usually use physical synthesis, for example, by laser ablation or ultrasonification, in order to reduce the size during the production of the nanomaterials. What I would like to point out today a little bit is that we have another alternative, which is based on the biological synthesis, in which we use or make use of organisms or of biological extracts of plants and other biological materials. And this is shown here. We have, a few months ago, we have published a review in which we distinguish between something what we call the pseudo-green synthesis and the real biogenic synthesis. The main difference is that on the left-hand side, if we have a look to this pseudo-green synthesis, that we use extracts from plants or from microorganisms or from biological fluids. These extracts will be incubated with metal precursors, and then we follow the chemical synthesis in order to produce the nanomaterials that we are looking for. The real biogenic synthesis is based on living organisms. So in this case, we take a living algae, bacteria, fungi, or yeast, for example. We put it into contact with the precursor again, and then the biosynthesis and the generation of these nanoparticles is performed within the organisms. The main problem is, and most people are using this approach because it's much easier, because you have let's say, an aqueous phase of this biological extract, and you have, just as a result of this synthesis, you will have a suspension of the nanomaterials within a liquid phase. However, in biogenic synthesis, you have the entire microorganism, and you need to find a way in order to extract these nanoparticles from the organism itself. And for this, I will give you a few examples later on. So, why are we so interested in biogenic nanoparticles? Because first of all, we have a huge variety of microorganisms that are able to produce these nanostructures, just putting them into contact with a precursor, for example, metal ions. And we have a huge variety. We have somehow the possibility to achieve the production of these nanomaterials by a green synthesis, because we minimize the use of hazardous chemicals. And why are they so interesting? Because some of them, of these biogenic nanomaterials, they show antimicrobial, anti-cancerous, or anti-inflammatory activities, and that's why they're so interesting for further applications. But how do you characterize these biogenic nanoparticles? And I will focus today in this webinar only on the core and not on the surface itself. So we have different microscopic possibilities, for example, transmission electron microscopy, atomic force microscopy, in combination, for example, with energy dispersive X-ray analysis. And the field we are working on is basically inductively coupled plasma mass spectrometry, and in this specific case, in the mode of single particle ICP-MS. The information you would like to gain out of it is basically the shape of the material, the size, which elements are involved, and of course the quantity. I won't talk about the quantity today, but a little bit about the shape and the size and the elemental composition of particles. And I will focus in this webinar on the use of single particle ICP-MS. In order to give you an overview how it works, I just show you the basic principle. In order, well, what we all know, if we're working with ICP-MS, if you introduce a dissolved analyte, you continuously introduce the analyte at a constant concentration. And if you introduce it like this, you will generate a relatively constant ICP-MS signal. This is just depending on the concentration of the element you have introduced into the plasma. If you use a suspension of particles, and the same is true if you use a suspension of cells, which I come later a little bit later to this. In this case, you introduce individual objects like this one, like this particle, and in the ionization source, you will form an ion plume that results in a very single and very short ICP-MS signal. So shown like here. If no particle is introduced into the plasma source, you don't have any contribution to the signal of the element. And once you introduce a particle, you will get a very short transient ICP-MS signal, like here, or the second particle, like here. And, well, these introduced particles, they produce this ion plume in a very, very short time. So the duration process is roughly half a millisecond. And in order to discriminate between various particles or also cells, you need to apply a very short integration time, lower than 10 milliseconds. It can go down to microseconds, for example. And what you do now is you use the analysis of very diluted samples in order to prevent that more than one particle is introduced to the ionization source at the same time. And you get the following information. Well, this is a typical experiment. In this case, it's the detection of phosphorus, in this case cells, but in particles it's the same. And you can see whenever a cell or particle is introduced into the ionization source, you will get this very short signal, what we call events. And you get a lot of information out of these experiments. The number of events, so the more events you detect, the higher the particle number concentration will be. The net signal of each event will give you the information about the elemental mass in the individual particle. For example, if we have this signal here, it gives a signal of, let's say, 150 counts. But in comparison to this one, which reaches almost 400 counts, means the signal was much higher. This means that in this particle, the element was in a higher, was present in a higher number. So the elemental mass was much higher. And then the third information you can get out of it is the background signal, which gives you the information about the element that is not bound to any particle or cells in this case. The first example I brought to you is about selenized yeast, and I will explain you why yeast. Selenium, of course, it's an essential element for humans, and you need to take a certain amount per day in order to have your body in a good shape. And of course, in some regions, the selenium or the food doesn't contain enough selenium, so you need some further added input of selenium that can be given, for example, by selenized yeast. So there are some commercial products, as you can see here on this side, which have up to 3,000 micrograms per gram of selenium per gram of yeast. So basically, the selenium yeast contains selenium in the form of the selenomethionine, so in an organic form. The inorganic selenium is relatively low, and the rest, well, can vary a little bit or is unknown. However, this study has been done in collaboration with the National Research Council of Canada, so in particular with Kelly LeBlanc and Salter Mester. And at that time, the PhD student Roberto was responsible for this work. He's now assistant professor at the Complutense University in Madrid. So why yeast? Coming back to this and checking whether they are intracellular selenium nanoparticles. Well, first of all, for microbiologists at least, it's quite easy to grow yeast. Seems to be very easy. I'm not an expert in this, but obviously it's the easiest thing of the world. And yeast as a microorganism is known to be a very excellent factory for any metallic nanomaterials, unless you do not intoxicate the yeast itself. However, there's a big problem. In order to get information about what is inside the yeast cells, inside the individual yeast cell, and this is the cell wall, which basically contains of a membrane and a second layer that makes this cell wall very rigid and it's very hard to let's say to lyse it, and this is a big problem. So how do you get access to any intracellular nanoparticles, or in this specific case to selenium nanoparticles? So in other case, how to extract these intracellular nanoparticles? For this, there was a very nice study from the University of Po, or from CNS in Po, of Joanna Spurner and Richard Lubinsky, and they compared 10 different extraction methods for the extraction of selenium nanoparticles from yeast itself. So they used one enzymatic approach, they used 6 different mechanical approaches and 3 chemical ones. And each of these approaches have pros and cons, as always. And for example, for the enzymatic ones, we have the highest mass recovery, or they observed the highest mass recovery. But however, they observed a much broader size distribution than the expected one. Another, let's say, disadvantage is that it takes almost 2 days to carry out this extraction procedure. As an alternative, they proved 6 different mechanical approaches, with the advantage that they basically provide a more realistic size distribution. So it's not as broad, for example, as in the enzymatic method. The time to extract the particles from the material is much shorter. However, the particle number concentration that you extract from the material is also much lower, which is of course a disadvantage. The other alternative would have been the use of chemical extraction methods. However, what they have observed is the present selenium nanoparticles, they partially dissolved. And this, of course, doesn't represent the original status or the original nature of the selenium nanoparticles in the sample itself. Here you can see a graph in which we have seen or where they show the different approaches on 4 different yeast samples. And on the one hand side, you see the mass concentration of selenium nanoparticles that could be extracted by these different methods. And basically here, the green one, the green column represents the enzymatic one, is usually basically much higher in comparison to the other approaches. However, if you see here on the, which is here the right one, the medium particle size, the medium one, not taking into account the distribution, you see that between the different approaches you have a relatively good agreement, at least for the one, let's say for the first 3 samples. For the 4th one, obviously the chemical method provides much higher medium particle sizes. So this reflects a little bit, although it's let's say a relatively simple methodology you apply for it and a relatively simple sample, that huge differences are obtained, and this of course requires more and more research in future in order to get an optimized extraction method, not only for selenium nanoparticles but for other materials as well. However, what we did, and this was somehow the reference method for mechanical extractions, what they used in the paper of Joanna and Richard, is a mechanical lysis of glass beads. How does it work? We take a certain amount of these glass beads, which have this diameter. We add to this the washed yeast, and then basically it goes some cycles of vortexing and ultrasonic treatment. After filtering, we usually apply two different methods, which is single particle ICP-MS and which is transmission electron microscopy. And well, we were quite content that the reference that the other groups, so an independent research and independent investigation, they showed that the size distributions observed, and we were basically interested in these size distributions, were in very good agreement with other methods. So according to them, and not only to us, the dissolution, the naturally being present biogenic nanoparticles do not get disrupted during the procedure using this mechanical lysis. So how do we check that the lysis has been of these yeast cells has been successful or not? And for this, we do not use single particle, but we use single cell ICP-MS. So here you see a typical example detecting phosphorus as a marker for an intact cell, because it is present in DNA, it is present in ATP and phospholipids and other compounds. So it's an excellent biome, or let's say a marker for an intact cell. So whenever you get a signal for this phosphorus, you can be sure that you have an intact cell. If you have selenized yeast, for example, you can see that you see also these events for the selenium. So this means that the selenium was incorporated into these yeast cells. So this was before the lysis, and then we apply the mechanical lysis. And what we observe is the following. In the phosphorus trace, you can see here in blue, you see an enhanced background, which means that the phosphorus from the cells has been released into the solution, but you do not see any individual events anymore. So this means that the disruption of the cell obviously was very successful. With alternative techniques, you can measure that it's around 99.5 or 99.8 of success, or let's say yield of cell lysis itself. Okay, in this case, we check the selenium, and you can see again a very similar picture like for the phosphorus. You see an enhanced background, but you don't see any events. So the conclusion in this case was that we believe that single cell ICP-MS can be a very nice proof of successful cell lysis because of the detection of phosphorus. But in this case, as there were no remaining events after the lysis, there were obviously no selenium-containing particles or particles that were below the detection limit of single particle ICP-MS. Let me show another example. In this case, we have used selenized yeast that has been incubated with selenite, so selenium-4. Again, the same picture. Before the lysis, you see the events of the intact cells. Obviously, they all contain selenium. And after the lysis, we see the following. The same picture for the phosphorus again, an enhanced background, very few events, which basically say that you have more than 99% of the cells destroyed and lysed. But the picture of the selenium is now completely different. There are remaining some events that indicate the presence of selenium-containing nanoparticles in the yeast. And in fact, out of these results, with some quantification approaches, you can detect biogenic selenium-containing particles in the size range between roughly 40 and 200 nanometers. But can we trust these results? For this, we always need a complementary technique. And what we usually use is transmission electron microscopy. And here you see some pictures of the particles we have detected in this yeast. So the same one we have analyzed before with single particle ICP-MS. And you can see a huge variety of sizes, but they somehow confirm the results we have seen in single particle ICP-MS. And basically most of them are spherical, let's say it like this, okay? Another example, the same corporation again, is we were interested in the detection of selenium nanoparticles in edible mushrooms. Again, the sample incubation and the sampling was carried out at the National Research Council of Canada, and these mushrooms were incubated with different concentration of selenite between 0 and 75 milligrams per liter. So we have two examples, the oyster mushrooms, which is relatively famous, at least in Europe. I don't know about in Brazil, and the linksy mushroom, which might be more popular in Asia. So these type of mushrooms we have analyzed. In this case, we did again the same lysis of the mycelia. In this case, the student who was responsible for this was Andres Suarez, who will defend his PhD at the end of the year. And he used a very similar mechanical lysis of the mycelium, again, using these glass beads, and again brought these suspensions to either single particle ICP-MS or to transmission electron microscopy. And what we observed is, at that time, we switched a little bit the experimental condition of our ICP-MS because we thought, well, these low transport efficiencies that usual nebulizer system can provide, this has to be a little bit switched. So we switched completely to now in our laboratory to low flow nebulizers in combination with so-called total consumption spray chambers, which are not working at 100%, but they can provide very high transport efficiencies of more than 70%. If you're interested in more information about this, last year we have published a comparison for different systems of these low flow nebulizers with different spray chambers, and we see two tendencies basically. First of all, the transport efficiency depends on the type of sample you're analyzing, not only the type of analyte, so whether it's a particle or a cell or whatever you're interested in, and it depends on the matrix of the sample you are introducing. So basically you need to check for each system you are analyzing, or for each sample you're analyzing, you need to check for these transport efficiencies, but in some cases you can get really high ones, up to 90% in some cases, and this of course gives you much more information about the sample in comparison to a system where you have very, very low transport efficiencies. Okay, the results what we observed, and this is a lysis of the mycelium, because in all the other parts of the mushroom we haven't found any particles, is as the following. Here is the mycelium of the linksy, independent of the incubation concentration. So here between 20 milligrams per liter up to 75 milligrams per liter. Basically, the size distribution of the particles we detected is very, very similar. And here, even better, shown for the mycelium of the oyster, it has very, very little variation, and usually independent from the species, independent from the mushroom, it is roughly in the range between 70 and 130 nanometers. And again, to prove this, we always take into account a complementary technique like the transmission electron microscopy, and it proved somehow very nicely the results of single particle ICP-MS. However, we want to go a step further. We want to know a little bit about the core composition of the selenium nanoparticles. We know by all the experiments that they contain selenium, but in which form? We were interested in to check for this. And here we made use of a very simple chemical reaction. The sodium sulfide reacts selectively with only elemental selenium to form this complex. So this means if we have particles present only containing elemental selenium and we put it into contact with this reagent, they should disappear, they should be dissolved because of this reaction. And what we see is before the treatment, we see the regular analysis, seeing the events of the selenium, of the present selenium containing nanoparticles. And after the treatment, you see that we have only a baseline. No events occur anymore. So basically, 100% of the present selenium nanoparticles in these samples were of elemental selenium. So this is an information ICP-MS, or an additional information ICP-MS can give as well in this specific case. Okay, in order to summarize a little bit this first part about selenium nanoparticles, we believe, and with the support of other groups, that the mechanical lysis of biological material is a very suitable method for the isolation of biogenic materials. We will see later examples that work not only for selenium particles, but for other systems as well. In general, single particle ICP-MS is a powerful tool for the detection and characterization of these selenium nanoparticles. However, the drawback is always the size detection limit. In the best case, at least with our instrumentation we have at the University of Oviedo, the particles have to be bigger than 20, or need to have a diameter bigger than 20 nanometers. And what is always needed is that you have a complementary technique in order to get an agreement between the results you obtained by ICP-MS or by single particle ICP-MS, because single particle ICP-MS just gives you information about the element, but not about the size, not about the shape, and nothing else. So you need a complementary technique. Okay. Let's switch to a second topic. I want to talk about some other food products, and this work has been carried out in collaboration with David Heath from the Joseph Stefan Institute in Ljubljana in Slovenia, and Ignacio Machado of the Universidad de la República in Uruguay. And these food products are the following ones. So these mealworms, the grasshoppers, or crickets. Yeah? You might be surprised about, or maybe you have tested them, but these edible insects, they are relatively famous in many regions of the world because they provide a lot of interesting ingredients, like high protein contents, essential amino acids, high content of fibers, polyunsaturated fats, vitamins and minerals, and so on. So to see these insects as a alternative food product can be very interesting in future, when you see that they can provide potentially positive impact on human health. So just to compare it a little bit, and we compared here the nutrients concerning the protein, fat, carbohydrates, and fiber between regular animal food, let's say it like this, between fish, chicken, beef, and pork, and compared to crickets or yellow mealworms, you see that they also contain very high protein content, very comparative to the other examples, very high, sometimes fat, but the percentage of unsaturated fatty acids might be very, very high. They provide some carbohydrates, and which might be not so unimportant, they provide relatively high concentration of fiber. If we have a look to the map here, you can see that there are many countries in which a lot of different insect species are consumed already. I think the leader, of course, is data of more or less 10 years old, but the leader is China. But you can see India, Australia, they have a huge number of different insect species that belong to the food. But also, for example, Brazil, they have, not sure which of these, let's say about 200 different insect species that can belong to the daily menu of the population. Of course, we are more interested in the metal concentration in these insects, and we analyze 4 different species. So the mealworm, the grasshopper, and 2 different species of the crickets, and we checked for some certain elements using just relatively simple microwave digestion, typical for organic materials or biological materials, combination of nitric acid and hydrogen peroxide, and then analyzed by ICP triple quadrupole MS. And I will focus just on 5 elements because we measured some more, but you can see that concentrations, for example, of zinc, they're relatively high. Selenium is present in all of these species. The concentration of iron is relatively constant, independent from the species you analyze. And of course, then you have some values relatively high for aluminium. And this, of course, can be of disadvantage because aluminium can be a toxic element or belongs to the toxic elements. But of course, it's not only important that the total concentration of certain elements are very high, but whether they are bioaccessible for our human body. And there are these standard protocols they can use, and you can imitate the oral phase of absorption, the gastric phase, and the intestinal phase. And we did the same with the different insects we have analyzed. And here are some interesting results, I would say, because zinc, for example, is basically close to 100 absorption or accessible for the human being. The availability of selenium is also very, very high. In the case of iron, it depends on the species itself. So there might be different iron species being present, for example, in the mealworm and in the grasshopper than in the cricket, because here the bioaccessible fraction is much, much lower, which is a very, very important factor. Copper is, well, it varies a little bit, but it's relatively high. But very important is, although these species have, or these insects have relatively high aluminium concentration, the accessibility is fortunately relatively low. So what we observe, or let's say as a conclusion, because we were basically interested in the elements iron and aluminium, that iron is basically very high accessible, at least in these 2 insects, and aluminium is at a relatively low rate. And we wanted to know whether particles are involved. So what did we do? We analyzed again the different extraction from the different insects of the species, and we analyzed, for example, here iron-containing particles by single particle ICP-MS. And guess again, Andres had to do this work, but very successfully. So what you can see on the first view, if you see the first experiments here, these signals, these individual events indicate the presence of iron-containing particles, for example, here in one of these crickets. The same is true for aluminium, but I don't show you the data. And if you have a look to the mass distribution of these 2 elements in the particles, you see, besides here this exception, they are almost identical for the different insects themselves. Here, in the case of iron, the masses of iron per nanoparticle varies between roughly 1 and 2 femtograms. In the case of aluminium, it varies between 0.1 and maybe 0.6 femtograms per nanoparticle, but basically independent from the insect itself, besides here in the case of the iron. And again, we need a complementary tool in order to get information whether the results of ICP-MS make sense or not. So in this case, we use the combination of high-resolution transmission electron microscopy and energy dispersive X-ray spectroscopy in order to image some selected particles. Here on the left-hand side, you can see some iron particles, which are not really nanoparticles, because you see they will have roughly a diameter of 250 nanometers. But the EDX data give us a very impressive picture that this particle is based on iron, and it's probably an iron oxide particle. In the case of aluminium, it's very similar as well. We have here a particle, a smaller one, which obviously does not contain any aluminium, but here we have not a spherical one, but another particle that contains a high concentration of aluminium. And this is a very nice indication and a proof that the results of the single particle ICP-MS somehow make sense. So in order to conclude this, from the nutritional point of view, maybe not from the aesthetical point of view, insects are rich in proteins, fibers, and other essential compounds, for example, minerals. So some essential elements like iron, zinc, and selenium, they have relatively high bioaccessibility, and they might be in favor for human nutrition in principle. However, what we observed that this bioaccessibility is basically influenced by the presence of particles, for example, for iron. The higher the concentration of iron particles, or the particle number concentration is, the lower the bioaccessibility. But I don't want to go into more detail, but my conclusion is for sure it requires more research in order to get more information about this, let's say, new source of alimentation for human beings. Okay, at the end, I would like to acknowledge especially our group and the University of Oviedo. This is a very actual picture. And well, since a few weeks or months, we have also a profile in LinkedIn and also in Instagram. I acknowledge very much the support of some institutions and some private companies. And last but not least, I want to come back to Oviedo, because we have, let's say, a long tradition to welcome friends and colleagues from Brazil. I haven't found many pictures, but at least I want to point out here, Juliana and Diogo, they have stayed for 1 year with us some 4 years ago, and I think they enjoyed the time very much. Most Brazilians won't agree that he is capable to prepare the best caipirinha in Brazil, but at least in Oviedo. So what I would like to express is that colleagues from Brazil are very welcome in our research group, and usually they come as known or as unknown colleagues, but they leave as good friends. And I think this is also very important, not only scientific collaboration, but to have a good personal contact to each other. So with this, I would like to finish, and thank you very much for your attention.